Who are the top researchers in longevity? Who should you trust? What have they worked on? There are a lot of influencers out there claiming to be longevity experts. They post blood work, supplements, fasting schedules, and screenshots of their VO2 max. Some of what they say is useful. A lot of it isn't. Some of it's dangerously wrong. One of the most common questions I'm asked is, so who should you trust? Ultimately, people need to start with critical reasoning. Does what this person say sound too simple or too good to be true? For example, is there really one superfood that can cure all of your ills? I can answer that for you. No, there isn't. Or does what this person say sound too extreme or self-aggrandizing? For example, saying they have the world record in slowing their aging, or they are the equivalent to a teenager when they don't look particularly healthy. It's your job to spot these extreme claims and filter them out as noise. So today we're going to walk through 12 of the most respected researchers in the field of aging. These are people who didn't just jump into longevity for the clicks. They've spent decades in labs, running trials, building models, and publishing foundational papers. These are the people whose work defines the contours of what we know and don't know about human aging. I know most of them personally and adamantly believe that these are the people that should be getting the attention and acknowledgement, not the influencers and not me for that matter. Each one brings a different lens. Some specialize in metabolism, some in genetics, others in epigenetics, redox biology, AI, or developmental reprogramming. What unites them? Rigor. They do the work. This is not an exhaustive list. There are dozens of others who could easily be added, but it is a solid start with some of the best. So let's go deep, one by one. Credentials, contributions, even controversies, if any. Here's who you should know and why. Let's start with Dr. Nir Barzilai. Barzilai is a physician and geneticist. He earned his MD from the Technion in Israel and did his residency at Yale. Today, he runs the Institute for Aging Research at Albert Einstein College of Medicine, where he also directs the NIH's Nathan Schock Center and Glenn Center on Aging. If you've heard of the TAME trial, Targeting Aging with Metformin, which was the first major human study proposing to treat aging directly with a drug, that's Barzilai's project. But his scientific legacy starts before that. Barzilai launched the Longevity Genes Project, a study of over 600 centenarians and their families. What he found flipped assumptions. Instead of perfect lifestyles, most centenarians in the study had average or even poor health habits. For example, smoking, drinking, and maybe little exercise. What they had were protective gene variants, especially in pathways like cholesterol metabolism, CETP and APOC3, and insulin signaling. People with these gene variants had higher HDL, better cognitive function, and significantly lower rates of cardiovascular disease, cancer, and diabetes, even into their 90s and 100s. In a field often focused on caloric restriction and pharmacology, Barzilai brought genetics front and center. But he didn't stop there. He co-founded CoBar, a biotech company developing mitochondrial peptides, a novel class of signaling molecules encoded in the mitochondrial genome. These peptides appear to play roles in metabolism, inflammation, and age-related disease risk. His lab also explored endocrine signatures of long life, like adiponectin, IGF-1, and other metabolic regulators that differ in centenarians. Across more than 360 peer-reviewed papers, Barzilai has helped connect the dots between metabolic function, genetic variation, and exceptional aging. But what really pushed him into the spotlight was TAME, the Targeting Aging with Metformin trial. TAME isn't about proving metformin is a miracle drug. It's about proving a broader concept, that aging is a modifiable risk factor, and that drugs like metformin, which already have a safety record, might help delay age-related diseases, not one at a time, but all together, which is the essence of longevity research. The trial design is novel. Instead of testing whether metformin prevents diabetes, it will track whether metformin delays multimorbidity, cardiovascular disease, cognitive decline. If it succeeds, it could push regulators like the FDA to finally treat aging itself as a clinical endpoint. That opens the door for a whole class of gerotherapeutics. Of course, metformin is controversial. Some studies suggest it improves lifespan in diabetics. Others show no benefit in healthy individuals, but there's data suggesting it might blunt exercise adaptations, especially in older adults. Barzilai himself is measured. He doesn't claim metformin is a panacea. He positions it as a first mover in a broader regulatory strategy. There's also critique around generalizability. Much of his genetic work has focused on Ashkenazi Jewish cohorts, which may not translate globally. Barzilai acknowledges that, but emphasizes that the pathways discovered, like cholesterol transport, insulin signaling, inflammation, are broadly conserved and actionable. Ethically, Barzilai is clear. Preventing age-related disease is not just desirable, it's a necessity. He draws comparisons to statins and vaccines, tools that altered the trajectory of public health. He believes aging drugs should be regulated the same way, and that we have a moral obligation to push forward. He's not trying to make people live forever. He's trying to extend the healthy period of life, the part where you can move, think, and contribute. So when people ask who's doing the hard translational work to get aging drugs into the clinic, Nir Barzilai is at the top of that list. Now, in full disclosure, I'm a member of the Longevity Biotechnology Association, which Nir Barzilai founded and leads. If you want to learn more, check out his book, Age Later. Moving on to another frontrunner in the longevity field is Dr. Steve Horvath. He didn't come from biology. He actually came from math. With a PhD in mathematics from UNC Chapel Hill and a Doctor of Science in Biostatistics from Harvard, he's one of the few longevity scientists who entered the field through the statistical backdoor. That background matters because Horvath's defining contribution was computational, the creation of the epigenetic clock. In 2013, Horvath published a single-author paper that changed the way we think about aging. Using DNA methylation patterns across tissues, he built a multi-tissue age predictor that could estimate the chronological age of virtually any human sample with a margin of error of only 3 to 4 years. It worked in blood, brain, liver, skin, across the entire body. That model, now called the Horvath clock, has become a cornerstone of biological age research. It allowed scientists to measure aging rate, not just lifespan, but the velocity of decline. And it had properties that felt biologically meaningful. Newborns have a clock age near 0. Stem cells reset to 0. And in disease, clock age often runs ahead of real age, what we call age acceleration. This mattered because for the first time, we could test interventions. We didn't need to wait 40 years for people to die. If an intervention slowed or reversed the clock, we had evidence that it might be biologically relevant. From there, Horvath went further. He developed second-generation clocks like GrimAge and PhenoAge, built in collaboration with researchers like Morgan Levine, who we'll talk about soon. These models didn't just predict chronological age. They predicted mortality, disease incidence, even time to death. Horvath's lab also built tools like WGCNA, Weighted Gene Co-expression Network Analysis, which clusters genes into modules to find aging signatures across omics data. His group has published on Alzheimer's, immune aging, and transcriptomic aging in both humans and mice. But like any new biomarker, the clocks have drawn skepticism. Some argue they're correlative, not causal. Others worry about technical reproducibility. Early versions of the Horvath clock could vary by up to 9 years on duplicate tests. That's a problem if you're trying to measure the effect of a drug or supplement. Horvath addressed this. He released noise-reduced versions and incorporated principal component analysis to strip out technical variance. Newer versions typically vary less than a year across replicates. This is a major improvement. There's also debate on what these clocks actually measure. Methylation patterns change for many reasons. Cell composition, inflammation, oxidative stress. Are we measuring aging itself or the body's response to stress and disease? Horvath's answer is pragmatic. If the clock predicts death and disease better than chronological age, it's useful. Whether it's a readout or a mechanism is secondary. He's also said openly that more work is needed, that we'll need 3rd and 4th generation clocks, better tunes, more interpretable, less reactive to short-term noise, and that multiple clocks will likely be needed depending on the application. Ethically, Horvath has taken a strong stance on open science. His clock algorithms are public. His code is shared. He opposes companies trying to privatize or overcharge for methylation tests. He believes aging clocks are a public good, not a proprietary asset. He's also cautious about how these tools are used. He's warned that insurance companies or employers should never be allowed to use epigenetic age data for decision making, and that consumers need context, not just a number. In 2022, Horvath joined Altos Labs, a company funded by Jeff Bezos and Yuri Milner, a private rejuvenation biotech, where he's continuing his work on aging clocks and reprogramming. He remains one of the most cited and respected figures in the aging biomarker space. If you're working on longevity and you don't understand Horvath's clocks, you're missing the scaffolding on which most intervention trials are now built. Now, moving on. If there's a scientist who has consistently pushed for empirical clarity and intervention-first thinking in longevity, it's Dr. Matt Kaeberlein. Kaeberlein did his PhD at MIT in the Garante lab, where he worked on yeast sirtuins alongside doctors David Sinclair and Brian Kennedy, both of whom we'll get to later. Kaeberlein specifically focused on SIRT2, one of the first genes shown to regulate lifespan in a eukaryote. That research laid the groundwork for the sirtuin hype that followed, but Kaeberlein didn't stick to hype. He stuck to data. In the early 2000s, he shifted focus from sirtuins to the mTOR pathway, and this was pivotal. Alongside Brian Kennedy, he showed that inhibiting TOR could mimic caloric restriction and extend lifespan in yeast. This set the stage for one of the most important pharmacological discoveries in geroscience. Rapamycin extends lifespan in normal, non-genetically modified mice. That paper, published in 2009 by the Intervention Testing Program, changed the field. Kaeberlein didn't stop there. He co-founded the Dog Aging Project, a longitudinal study tracking thousands of companion dogs to identify aging trajectories and to test interventions like low-dose rapamycin. Why dogs? Because they age in a human environment. They share our homes, diets, even our diseases. They offer a way to test geroprotectors in a real-world context. They have more translatability than mice and more tractability than humans. And most importantly, because we love man's best friend. Kaeberlein's work is characterized by pragmatism. He doesn't chase headlines. He wants interventions that work, that can be replicated, and that can eventually be used in people. He's also a vocal critic of pseudoscience. In 2023, when another high-profile researcher claimed a supplement had reversed aging in dogs, Kaeberlein publicly called it a lie. He even resigned from a longevity research consortium to protest, emphasizing that scientific integrity must come before marketing. His lab has also tested alpha-ketoglutarate, showing that it can modestly extend lifespan and compress morbidity in mice. Early human data suggests it may reduce biological age. Kaeberlein's legacy so far includes foundational mTOR longevity research, pioneering rapamycin trials in mammals, launching the largest canine aging study in the world, and advocacy for the geroscience hypothesis that targeting aging mechanisms delays all age-related diseases. He's cautious with claims, clear with data, and relentless about testing ideas across species and models. If you care about intervention-driven science, Kaeberlein is one of the most grounded, no BS voices in the field. And I'm proud to say that Matt has been advising me and Novos since the start, including on our Novos Core formulation. You can find his podcast called OptiSpan and many interviews of him with big podcasters like Dr. Peter Attia and Tim Ferriss. Subscribe to my channel to see my recent interview with Matt that will drop soon. Moving on to Dr. Vadim Gladyshev is what you'd call a systems thinker, but with hard biochemical chops. He's a professor of medicine at Harvard and director of the Center for Redox Medicine at Brigham and Women's Hospital, and he's published over 400 scientific papers. Early in his career, he mapped out the entire selenoproteome, identifying the human genes that encode selenocysteine-containing proteins, which are deeply involved in antioxidant defense. That's important because redox imbalance is one of the upstream disruptors of protein folding, DNA stability, and mitochondrial integrity. These are mechanisms of aging. But Gladyshev didn't stay in oxidative stress. He moved into comparative genomics of longevity, asking, what do the genomes of long-lived animals look like compared to short-lived ones? He sequenced and analyzed the naked mole rat genome, the bowhead whale, and others. His team found that long-lived species tend to upregulate DNA repair, suppress growth pathways, and resist cancer. Those genetic signatures offer insight into what slows damage accumulation over time. Then came one of his biggest contributions, the discovery that biological age resets to zero in the early embryo. When a sperm and egg fuse, they carry years of biological wear. But within a few days, epigenetic age drops sharply. The embryo is rejuvenated. Gladyshev showed this by applying methylation clocks, remember Horvath, during embryogenesis. He identified what he calls the ground zero of aging, the point at which the embryo becomes biologically the youngest it will ever be again. This has massive implications. It suggests that rejuvenation is not hypothetical. It happens in every new human life. The goal now is to figure out how to emulate that reset in somatic tissues. It also indicates that aging technically starts at the fetus stage. Gladyshev is also working on partial reprogramming, redox signaling, metabolite-based clocks, and longevity signatures across mammalian species. He's actively building tools to predict maximum lifespan from genomic features, a cross-species map of the aging rate. No scandals, no hype, just deep, fundamental insight into what life is doing across evolutionary time and how we might hack it. Now for Dr. Brian Kennedy, who we mentioned earlier. He trained at MIT in the Garante lab, which was also the same origin point as Kaberlein and Sinclair. His early work was on sirtuins and calorie restriction in yeast, but over time, his lens widened. Kennedy helped show that inhibition of mTOR, not just sirtuins, could extend lifespan. This shifted the narrative from NAD metabolism to nutrient sensing and helped bring rapamycin into focus as a geroprotector. He later ran the Buck Institute for Research on Aging, where he promoted translational approaches to aging, including compound screening, aging clocks, and health span metrics. Kennedy also helped test alpha-ketoglutarate in mice, with results showing modest lifespan gains and compression of morbidity. He later co-authored a study in humans using AKG plus vitamin A, showing an 8-year reduction in biological age according to methylation clocks. That study made headlines, but Kennedy was careful. He framed it as promising, not conclusive. He's now based at the National University of Singapore, running aging trials, exploring combination therapies, and trying to bring aging interventions into routine care. Kennedy bridges the gap between basic science and human testing. His commitment to rigorous research is widely respected. He doesn't overclaim. He does the work, and he's one of the few scientists with real experience across yeast, mice, and human trials. Brian travels the world speaking at longevity conferences and bringing a fresh perspective on the field. While I'm fond of Brian, I'm not so sure how he feels about me after I stole Dr. Diogo Berardo from Brian's lab to lead up Novos's R&D arm. Next, we have to highlight Dr. David Sinclair, who is probably the most famous scientist in longevity and the most polarizing. He's a professor of genetics at Harvard Medical School, where he runs the Paul F. Glenn Center for the Biology of Aging. His early work focused on sirtuins, especially SIRT1, and their dependence on NAD+. He helped demonstrate that sirtuins regulate stress resistance, metabolism, and possibly lifespan, at least in model organisms. This led to the development of resveratrol as a SIRT1 activator and the founding of Sirtress Pharmaceuticals. That company was bought by GSK for $720 million in 2008. A few years later, enthusiasm cooled. Sirtuin activators weren't performing in trials. Sinclair stepped back from the company, but he didn't step back from the field. Instead, he shifted focus to NAD metabolism, DNA repair, and epigenetic reprogramming. His lab showed that NAD plus precursors like NMN restore vascular and metabolic function in aged mice. These results contributed to the current NAD booster craze. Sinclair also introduced the idea that epigenetic noise is a core driver of aging. In 2020, he published a high-impact study using ICE mice, in which DNA double-strand breaks accelerate epigenetic aging. He showed that this process is reversible with partial reprogramming using Yamanaka factors. That's a big claim, that aging isn't just damage, but a loss of information, and that we can reinstall that information. Sinclair's public profile exploded with his New York Times best-selling book Lifespan and his media appearances. He's been criticized for overhyping, for example, implying we already have drugs that reverse aging in humans. Some researchers argue his optimism outpaces the data. Still, Sinclair is undeniably influential. He helped reframe aging as a reprogrammable system, not just entropy. And he's catalyzed interest and funding in areas like NAD biology, mitochondrial function, and reprogramming. He was also an inspiration to so many young scientists entering the field, so he can be credited for building a lot of the excitement and attention that longevity is now getting. I met with David at his Harvard lab in 2019 when Novos was nothing more than a concept. David was a big fan of our approach and plan, and along with Dr. George Church, gave me the validation I was looking for to decide to launch the business. And for that, I am grateful. Now, speaking of George Church, let's talk about him. Dr. George Church is the polymath of the aging world and biology. He's a geneticist, molecular engineer, and synthetic biology pioneer. He's a distinguished professor at Harvard and MIT. co-developer of CRISPR. His lab has spun out over 40 companies. Church's work spans from genome sequencing to gene therapy, but in aging, his focus is genetic reprogramming, genomic stability, and gene delivery platforms. He's proposed that aging is not just modifiable, but reversible, and not over decades, but potentially in a single shot using multi-gene therapy. In 2020, Church's lab published a study delivering 3 longevity-associated genes, FGF21, alpha-klotho, and TERT, via adeno-associated virus in aged mice. Result? Lifespan extension, improved muscle strength, and reduced frailty from a single administration. This points toward what Church calls a gene therapy cocktail for rejuvenation. He's also investigating epigenetic reprogramming, telomerase delivery, base editing to remove somatic mutations, and even gene drives to spread protective variants in population. What makes Church unique is that he doesn't just propose ideas. He builds the tech to execute them. His lab created multiplex gene editing, cell lineage tracing, 3D genome mapping, and a lot more. His innovation, inventing direct genome sequencing back in 1984, eventually brought down the cost of genetic sequencing by 200,000 times. Truly legendary. In aging, Church's vision is clear. Use gene therapy to reprogram human cells, restoring function, repairing damage, and eventually turning back biological time. His roadmap includes using gene circuits to make reprogramming controllable, tunable, and safe. Church has been critiqued for proposing radical ideas like resurrecting extinct species, which he recently did with the dire wolf and intends to do with the woolly mammoth. Scientifically, his work is rigorous. He's not a biohacker, he's a builder. And if gene therapy becomes a cornerstone of longevity, it'll be on platforms Church helped to invent. I owe a debt of gratitude to George for joining our scientific advisory board when Novos was nothing more than an idea back in 2019. Subscribe to my channel to see an interview that I recently filmed with him at his Harvard lab that will drop soon. Another pioneer in the world of longevity is Dr. Juan Carlos Belmonte, who is one of the few researchers who has reversed aging phenotypes in living mammals. Formerly at the Salk Institute, now at Altos Labs, Belmonte's core expertise is developmental biology. He studied limb regeneration, embryogenesis, and tissue identity for decades. In 2016, his lab published a study that changed the game. In a mouse model of progeria, cyclic expression of Yamanaka factors reversed signs of aging and extended lifespan. This was the first in vivo demonstration of partial epigenetic reprogramming as a rejuvenation tool, rejuvenating organs. The treated mice had less DNA damage, better organ function, and longer lives without erasing cell identity. Belmonte then showed that short pulses of cyclic expression of Yamanaka factors in wild-type old mice improved muscle regeneration, brain plasticity, and pancreatic function, again without triggering teratomas or dedifferentiation. This work gave legitimacy to the idea that epigenetic reprogramming could be safe and therapeutic. More recently, he's been engineering delivery systems using adeno-associated virus vectors, drug-inducible expression, and tissue-specific promoters to make reprogramming controllable in vivo. He's also worked on chimeric organ generation, including human-pig hybrids for organ transplantation, and embryo mapping using single-cell sequencing to chart aging and development. Critics warn of tumor risk and loss of identity with epigenetic reprogramming. Belmonte's answer? We're not doing a full reset. We're rolling back just enough to restore function. If partial reprogramming, making aged organs young again, becomes real-world medicine. Moving on to J. Pedro de Malgajais, he is a data-first biogerontologist. His focus, genomics, databases, and cross-species comparisons. He runs the Integrative Genomics of Aging group at the University of Birmingham. His name is behind many of the field's go-to public resources. GenAge, DrugAge, which was created by Novos' own Dr. Diogo Berardo, AnAge, and CellAge. These databases are what catalog genes, drugs, cell lines, and phenotypes related to longevity. He's published on computational prediction of longevity genes, network analysis of aging pathways, and biomarker validation frameworks. He's also sequenced genomes of long-lived species like the naked mole rat and bowhead whale, contributing to the search for conserved anti-aging adaptations. More recently, he's explored AI for geroprotector discovery and gene expression atlases of aging tissues. His work bridges bioinformatics and translational biology, especially for target discovery and biomarker development. Malgauhais is also a thought leader. He's written policy papers urging regulators to classify aging as a modifiable condition and pushing for more centralized infrastructure to support geroscience. No controversies, no drama, just reliable, well-curated data, and a belief that with the right tools, we can decode the aging process algorithmically. Pedro has been an advisor to Novos from the very start, joining our SAB back in 2019. It's thanks to Pedro that we have Dr. Diogo Berardo on our team leading up R&D, and I'm grateful for that introduction. Now for a refreshing researcher in the world of longevity. Dr. Morgan Levine is one of the youngest but most impactful scientists in the field. She co-developed PhenoAge and GrimAge, two of the most predictive epigenetic clocks available. PhenoAge uses DNA methylation to approximate physiological age based on blood biomarkers. GrimAge goes further, predicting time to death, disease incidence, and even smoking history. Trained in demography and gerontology, Levine brings a rare combination. She understands population-level risk and molecular-level signal. Her work links epigenetics, transcriptomics, proteomics, and single-cell data to aging phenotypes. She's especially focused on how reprogramming resets aging and how to model that shift computationally. Now at Altos Labs, she leads AI-driven efforts to map aging and rejuvenation trajectories at single-cell resolution. Her goal? A molecular atlas of how cells age and how they unage. Levine is also an advocate for biomarker transparency. She's cautious about overclaiming clock data, vocal about test reproducibility, and regularly involved in validation studies. She's pushing aging science into the multi-omic, data-integrated future where aging is no longer just visual, but quantifiable and trackable. While her work has already made a big impact on the field, keep an eye on her because she's only getting started. Next is Aubrey de Grey. Aubrey de Grey is one of the most controversial figures in longevity and also one of its earliest visionaries. He proposed the SENS framework in the early 2000s, 7 categories of damage that drive aging, each with a corresponding repair strategy. The approach was bold. Remove senescent cells, fix mitochondrial mutations, clear extracellular aggregates, break crosslinks, and do it in parallel. De Grey was early on senolytics, mitochondrial gene rescue, and immune rejuvenation. SENS didn't invent these fields, but it helped fund them before they were fashionable. He was a provocateur. He said the first person to live 1,000 years might already be alive, that we could reach longevity escape velocity by the 2030s. Many scientists called this science fiction. Some respected him, others dismissed him. But the truth is, much of what he proposed has entered mainstream longevity research, albeit with more caution and nuance. De Grey now runs the LEV, or Longevity Escape Velocity Foundation, continuing mouse trials of combination therapies. De Grey's influence on public awareness is undeniable. He reframed aging as a problem that could be solved, and his work was eye-opening for me when I first came across the field more than a decade ago. Finally, we cannot forget about Dr. Eric Verdin, who is a physician scientist and president of the Buck Institute for Research on Aging, one of the most respected aging research centers in the world. Verdin's background is in immunology and metabolism. His lab helped define how sirtuins, ketone bodies, and NAD+ regulate cellular stress responses. He showed that beta-hydroxybutyrate, which is a ketone produced during fasting, is more than just fuel. It acts as an HDAC inhibitor, modulating gene expression. His group also demonstrated that SIRT3 regulates mitochondrial metabolism during caloric restriction. Verdin has explored how metabolic states, fasting, ketogenic diets, NAD+ availability, impact inflammation, immune cell phenotype, and resilience. He's a cautious, data-driven communicator. Where some researchers hint at miracle cures, Verdin emphasizes health span, quality over quantity. He frames longevity as maintaining function, not escaping death. Under his leadership, the Buck Institute has expanded its translational focus, partnering with industry and launching biomarker trials for aging. No scandals, no exaggerations, just rigorous science, collaborative ethos, and steady leadership. So, who do you trust? Start with the researchers who run labs, who publish, who share their lab data, who acknowledge limitations, refine their models, and keep showing up to do the work. Longevity isn't about outlandish claims. It's about reproducibility, about mechanisms, not just metrics. years. The scientists we covered today, Barzilai, Horvath, Kaberlein, Gladyshev, Kennedy, Sinclair, Church, Belmonte, Malgajes, Levine, DeGray, Verdun, they don't agree on everything, but together they define the scaffolding of the field. If you want to understand what's real in aging science and what's just branding, this is where you begin. So thanks for being patient and sticking with me. Stay sharp, stay evidence-based, and always be a little skeptical. Until next time, slow your age.